brain that is on the slides. Okay, so look at all the brain slides. Know them as nervous tissue or brain. The structure of the nerve cell, of the neuron, according to the dendrite. So if you look in your lab manual and you see all the little structures for each tissue, yes, you are responsible for knowing all of those structures. So if you haven't looked at the slides like that before, you've got a week to do it. All right? The other thing she can do is use the model. This is the neuron. Be able to identify at least the dendrites and the axon. The rest of it, maybe not so much. I mean, if she opened it up, yeah, there's a nucleus. You'll be getting this again when we get to sensory and motor. Smooth muscle. So you need to be able to identify this as smooth as opposed to skeletal, as opposed to cardiac. Within cardiac, you have intercalated discs that you need to know. Okay? You don't need to know the function right now, but you do need to know them as intercalated discs because they are on your list, or are on the list of things to know on your slides, for that slide. This one is simple columnar. Now if you notice, sometimes I have the labels taped over, sometimes I do not. If I do not, use that information, okay, to help you answer it. That's where knowing your chart, the big old chart that she gave you with all the tissues, all the functions, that comes in handy in knowing, oh yeah, smooth muscle is in the small intestine. Or this one was pancreas. Okay? There are ducts in here that are cut longitudinally. There are ducts in here that are cut on cross-section. I put it on the cross-section. Also know the other big spaces you see in there are also ducts, but they're cut in a longitudinal. Anytime you have a duct, the center portion is always the lumen. All right? The thyroid. For the thyroid, I just put the pointer on the lumen. I didn't do parafollicular for you, but you do need to know that. You should know that this is the thyroid. So when we did endocrine last week, you should have looked at all your endocrine organs. This is very distinctly different from the pancreas. It's distinctly different from the adrenals. It's different from the pituitary. Any one of those endocrine organs she can give you and ask you not only what the organ is, but what is the layer, what specific tissue, what specific cell types do you have. Make sure you read the question. If she wants tissue, give her tissue. If she wants cell type, give her cell type. Right? I can give you partial credit there, but I can't give you the whole thing. These are two types of scales. We use this one. There will be something here for you to weigh. So you will put it on there and you will weigh it. This we didn't use, and technically I could hold you responsible for it, but I'm not going to. There's a whole website on how to use this scale. It was used in previous semesters. I'm going to ask her if she really wants you to use it, but I'm not going to hold you responsible for it. All right, these guys. So you need to be able to identify each of these specifics and exactly what it is. This is a graduated cylinder. Any one of these. This is a beaker. Okay? This is a micropipetter. Yes, you need to be that specific. These are the tips for the micropipetter.
We just call them tips.
You can call them tips. Skin model. All right. So this is the sebaceous gland. I'm sorry I had it on the wrong thing. It's going fast. But you do need to know the layers of the epidermis. Right? You do need to know what is inside of the dermis. So all of these receptors, the glands, the hair, the muscle, you need to be able to identify all of that in there. And then this is the hypodermis. And look at where the Pacinian is. It's pretty much in the hypodermis, whereas your light touch is in the dermis. It's in the papillary layer of the dermis. Remember, you have papillary and reticular. You have two different types of connective tissue in there. You do need to know that, right? That is in your chart. This is where you're going to use your chart to help you identify what's in each tissue. It gets that specific. Okay, mitosis. You'll be using model and slide. Whenever you see condensed chromosomes, you know that you are in mitosis. If the chromosomes are not condensed, that would be interphase. We don't have an interphase model. We did out of nowhere when it kind of walked away.
Go in front of me?
Right?
No, it's fine, thanks.
These are both prophase. This is early because the centrosome is still intact. Both centrioles are together. This is late because the centrioles are starting to migrate to the poles, in which case you're going to attach to the centromere of each chromosome and start pulling them. Okay, but this is early, this is late. Now, this is another identification of your equipment. Most people get this wrong. What is it?
Transfer
A transfer pipette. I need both words. Transfer and pipette. You give me pipette, I give you half credit. Right? What's this stuff?
Parafilm.
Parafilm. Don't need the wax, just parafilm. Alright, don't call it saran wrap. This little tiny thing is a microtube. Okay, don't just put tube. Be able to distinguish between a cuvette and a test tube. The cuvette has the line. Okay, it's what you're going to see up there, kind of a clue. That's the cuvette. This is a cuvette. This is the container for it. What was in here, and you do need to look at the whitefish blastocysts. This is an easy one. This is perfect. But for each phase of mitosis, you should be able to recognize it. I have a PowerPoint that I'll be showing you, and I'll show you the phases. I'm sorry number 20 wasn't here, but it's the whole gland. This is an ovary. Okay? 23 is on a primary follicle. What is ovulated is a secondary oocyte. Remember, you do not finish meiosis until fertilization, which means if the secondary oocyte is flushed, you never finish meiosis. There's no reason to expend that much energy to do that. Okay, how many of you have done the calculation?